Is that okay? Good. Now we are in the panel view. So thanks everyone once again for your excellent presentation, and learned a lot from the TCP and the funding structure, and also the situation in Vietnam and Thailand. And I think there's been a very strong momentum to be really inclusive and to achieve the level of equity that we desire to be a part of the Human Cell Atlas. Perhaps I think Hyung-Suk asked this question earlier to Jonah, but I would love to open it up to other speakers. And this is a question that Christine Wells asked about, how's the research funding interest in terms of basic research versus the disease? Because I think there's been a huge kind of a strategies transformation within the funders to focus our research into more translational, but Human Cell Atlas being more basic science. Is there a potential conflict, or how are you overcoming this kind of question? So we'd love to hear this from Varadum or Nam from your side. So maybe Varadum, can you start us off on your thoughts?
Right, yeah. I don't think I have any good answers just to start with, sorry. But yeah, I think that's sort of the problem, like, that we see everywhere in the world, right now. So so I think even now, I mean, for IDA, we focus mainly on healthy samples, right? But I think I mean, to go around this, there's one way you can do it. It's just that, it's at least in Thailand, I think it's a lot easier to get the funding if you focus on one particular disease or something like that. So what you can go around is, which is, I'm not saying like this should be like a long-term solution, is that you can use the like fundings like from CCI to support like healthy samples and as a reference and so on. So you always can make a case for that, and also you can also apply for local fundings on something or diseases that really important for your own countries, and in a way, I think you can always make them understand that you can use you can combine the data and use the IDA sample as reference or as control or something like that. So I think you can always try to do something in the meantime. But for long-term solutions, I agree that in the end, it has to be like the bigger pictures and long-term solutions that can cover not only just certain diseases, and but also like the healthy sample as well. So I don't know what Mom thinks about like the situation in Vietnam. Is it something similar?
Yeah, I think I agree with Baradom that something similar in Vietnam. So Vietnam is kind of new to some research like single cell sequencing analysis. So first is the funding. The funding agency now focus on some other domain, and not so far. Even in biology, people is not aware enough to the single cell sequencing and the benefit from doing project using single cell sequencing technique. So I think there are two things. The first is to get the funding agency interested in funding those just projects using this technology. Maybe HCA may need. We may need HCA to raise awareness for some funding agency in Vietnam. Even in our institute, Big Data, we have also Innovation Foundation. But through 2 or 3 years, there's no project submitted to the foundation. And the second thing is that the researcher in Vietnam, even right now, I think, I don't know everything that happened in Vietnam, but most of them they focus more on some clinical aspect that can be applicable in Vietnam in this current situation in Vietnam right now. And single cell sequencing and also the single cell sequencing technique, they may not find something that is directly applicable to their clinical problem in Vietnam currently. So those maybe also need our awareness, our speaker to raise that awareness in Vietnam to have a long-term perspective with the technique in Vietnam.
No, excellent point that you raised. But I just wanted to touch upon your presentation that you have some collaboration with pharmaceutical companies, or this drug profiling you're doing with sequencing and genotyping. So may I ask how that project is funded?
Yeah, so we have several main projects here. So for disease risk prediction, we have internal funding project, and we also have a funding from VinIF, the Innovation Foundation of Vingroup as well. So for adverse drug reaction project, we also have some funding from national agency in Vietnam named some named I don't remember exactly the section that they fund, but at least they have some biological and biomedical section that they fund the project. But it's limited funding, and for for single cell sequencing, it's take a lot of money, and it's not just something that some national funding agency can affordable now. So we most of them we have the internal funding from the Vingroup Corporation, so they invest on some long-term projects.
Great. I think maybe others can.
Yeah.
Yeah,
So there was a question from Al, which I thought, like the data. I mean, like a lot of the countries are not actually aware of the power of this data sharing between networks and the countries. So as Al suggested, maybe there the securing funding body. Like for basic science for single cell research, or having the training grants for those who are interested in countries like in Thailand or Vietnam to develop their single cell genomics data and share the data together in the network in HCA or to the public would be really, really valuable. So maybe putting some efforts into securing some kind of a non-profitable funding body or developing a training grant would be maybe necessary. That's what I thought while hearing some of the great talks today.
Good point. Partha, I think you wanted to say something. So
yes.
go ahead.
The first thing, I'm going to keep my video turned off because I'm traveling in a car right now. So I'll keep my video turned off, but I do want to make 2 or 3 points. Number one, I've been championing the HCA in India. The major funding agency that funds all of this kind of research is the Department of Biotechnology, Government of India. We actually do not have much of private funding, hardly any private funding for research in India. So it's mostly government funding, and the Department of Biotechnology is essentially the biggest funder. And I've been championing with them. What the Department of Biotechnology, again, I was a part of that, spearheaded that in some ways, has come up with a national document on data sharing, which is really open data sharing, as long as privacy and confidentiality issues are not, are actually accepted, and appropriate precautions taken so as not to divulge details, identifiability issues. The data can be shared anywhere. It's just that the data also has to be retained. A copy of the data has to be retained in the country, but it can be shared for research purposes. When it comes to commercial use, then appropriate approvals need to be taken. But for research, this national document that we have prepared allows for open data sharing. Also want to make one last point is that since I've been pushing HCA or pushing towards obtaining some kind of exclusive funding from the Department of Biotechnology for HCA research in India, one of the things that the government does not want to hear is if it's not tied to some disease, they do not see the benefit, or they do not readily see the benefit, because this is expensive research, and they say that ultimately it must impact on some disease of national relevance. And so it's been a little difficult to obtain not a little difficult, it's been very difficult to obtain funding just for healthy controls without any reference to any disease or patients. So our group presented this morning, we are having to do this under the guise of oral cancer. And I say under the guise, not because we are not interested in oral cancer, but if we didn't have to sample patients, we could actually have increased our sample size, which would have mattered. So, but we can't get funding to do just healthy controls. So it is important that, in India, if you're wanting to generate national funding, it has to be in the context of a disease. Thank you.
Thank you, Partha. And yeah, we understand the importance of tying the question of disease with healthy control, perhaps as a package, rather than proposing a single project that only involves healthy tissues. But maybe I'd like to throw this question to DCP on the point that Partha also raised in terms of data sharing, but also Alistair asked the question earlier whether there is a possibility to let me find the question again, to distribute managed access. And I guess what I mean by that is if the data cannot leave the country, whether there will be a way to provide a distributed form of managed access.
Right. So can you hear me?
Yes.
Okay. Yeah. So there is a plan for being able to host data in different locales. So the underlying cloud platform does allow that capability, and we're going to exploit that to allow certain managed access data sets, if they have to stay in a certain locale, we'll be able to do that.
So maybe related to this, I guess, or you already provide ways for users to access expression tables. Is that right? Is that feature already available for people to use?
Yeah, we provide access to expression matrices, to the count matrices, cell by gene matrices. Yes, we both submitted by contributors and generated by the DCP pipelines.
Great. Okay. I think Varadum has a question.
Just a quick one to DCP, if that's okay. So right now, if I'm not mistaken, right now I think DCP's stored data is mainly from HCA, right? So that means mainly the healthy samples. So I wonder, it's going to take quite a lot of effort, but is there any plan that, if there's something similar to what I mentioned, like, if we have healthy samples that funded by HCA, for example, but there's a matched samples that, sort of disease group or something like that, would there be any possibilities that you can link, the two, the disease samples together with the healthy sample in some way? If that question makes sense.
So I can kind of field that one. So we actually do have disease samples inside the DCP. While the whole the HCA as a whole is trying to obviously make that healthy human reference, we do realize that a lot of, I think as Partha said, a lot of it is based on disease research, and it can be very difficult to find just healthy donors for different organs. So we do combine, and we do link it, and we do have fields in our metadata for diseases, for at least a donor with diseases or the actual tissue with disease. So we do take in that data.
Thanks, William.
Maybe related to funding as well, and I think it was asked by Irene earlier about infrastructure being, perhaps there are two types of infrastructure. One is the data production, like running 10x genomics or running single cell RNA sequencing, and which can be somewhat costly, but you also have another infrastructure of database and computation analysis. And I think DCP provides this type of access and ability for people to access and analyze. Maybe I'll ask the question to Partha or Varadum or Nam, or even Joshua's back online. what is the emphasis in your local research community of putting computational and bioinformatics into your program? Because I think that really is the future in a way that there's just so much data, wealth of data out there, yet there's untapped opportunities. And could that be something that to build upon, of course, in addition to data production, but at the same time, computational development. So maybe Joshua's here, and maybe I'll have Joshua speak to us first, and then I'll later introduce you for your talk. But
yeah, thanks everyone. So sorry, apologize for the incident. Anyway, in Hong Kong, I think I obviously can't speak for all of Hong Kong, but I think in Hong Kong we actually have quite a lot of very separate sequencing effort for single cell RNA sequencing. So people studying cancer, studying different kind of diseases or tissue. So they're quite separate effort. And I think bioinformaticians are increasingly playing a major role. So I came back to Hong Kong about 3 years ago, and since then we have got more and more bioinformaticians who really specialize in single cell analysis. I think we are actually playing an increasingly central role and not only analyzing individual single cell data set, but combining them into some larger efforts. So I think, yeah, we are definitely heading towards this direction in Hong Kong.
Nam, any thoughts on Vietnam?
Yes. So yeah, I don't know many things in Vietnam because I just come back 2 years ago. But at least during my my time working in Vietnam here, so I think that right now researchers in Vietnam is more interested in computational aspect of biology. I mean, that they will focus more on, for example, our project here. When the first time I come back to Vietnam, it's difficult to get some people that are interested in computational pipeline development. But right now, it's more and more people that are interested in that. So computational development of single cell bioinformatics right now maybe a little bit limited in Vietnam in terms of human resources. But I think in next several years, it may be improved. For example, in our project here, so we have some intensive and some also powerful computational infrastructure, and those maybe it's okay to use them to do single cell sequencing data analysis. And also one special thing is that we can collect a lot of sample from disease and even adverse reaction of patient in Vietnam, and those sample can be so the single cell sequencing technique can be applicable for those sample. And I think I can raise some awareness in Vietnamese researcher about the benefit of using this technique for some long-term project, for some long-term benefit. So in terms of computational development in Vietnam, I think right now, even in some university in Vietnam, they also interested more in training of bioinformatics, the domain of bioinformatics, and that's maybe a promising things we can see in Vietnam.
Great. Maybe if before Vardum, Jonah, you mentioned this very recent RFA that's currently open call, which allows more computational analysis to explore the atlas. Could you expand a bit more on that RFA, and is there a training component within that RFA?
Yeah, so I'm happy to expand more upon it. So it's entirely focused on computational questions. So that's one thing maybe just to emphasize. So there's no data generation component to that. Really looking to kind of develop tools, benchmark tools, curate data sets that would make it easier to reuse those tools. So the training component is that within this RFA, maybe similar to others, is we imagine kind of helping to support a community of practice. So we would imagine that through the cycles of these RFAs, a number of different groups, maybe with varying skills or areas of expertise, will come together. And by kind of building that community, there will be an opportunity to kind of cross-pollinate and also build some expertise. So it may be that there are experts that know a ton about a specific biological system and have some computational expertise, within that same cohort as someone that is, you know, an expert in neural networks and machine learning with relatively little biological expertise. And by kind of bringing together similar or different challenges, I think there'll be a really great opportunity. The other thing that I'll maybe quickly mention is that, you know, reproducibility, and to make sure that the interoperability of the tools that are developed are shared via, you know, open source code, via, you know, Jupyter notebooks, to hopefully make it easier for others to use those tools is important to us.
Right, excellent. That's very important. And probably if there are audiences or the participants in the group that wants to engage and collaborate, maybe with you know, other countries, I highly encourage you guys to actively engage and contact the people in this community. Bharadam, maybe do you want to? Can you comment a bit from bioinformatics efforts in Thailand?
Sure, maybe a quick one. So as a a systems biologist and bioinformatics student, this is something quite very close to my heart. So Jay, I see it this way that pre single cell, I think like it's kind of like golden era for people who do computational analysis, because obviously you don't need sophisticated and expensive machines. So I think when you start working in let's say developing countries, I think if you have like powerful computers, you can start doing that right away. And in fact, you can share the facility with people from different disciplines like engineering and astrophysicists who also need HPC. So I think that's pretty straightforward. However, when you have a single cell, it becomes like previously that there was no problem with data generations because you have lots of RNA-seq, ChIP-seq, I mean the bulk one available. But right now, I think we start to have some shortage in single cell data, at least in Thailand, because I guess it's still quite expensive to produce the data. But eventually, I think it would become cheaper and cheaper, and we would face the same problem as any other omics, because that you have too much data and you can analyze. And my last point would be that you can hire people who are in IT and something like that to try to do programming for you. But in the end, I think the people who can really analyze the data would be someone who have to have the technicalities to do programming and also understanding the biologies, and sometimes it's quite hard to train those people. So I really agree that we need to improve the global effort to try to train people who can do this properly.
Yeah. That's exactly what Jonah's point of, if you could find a good collaboration with biology and computational, that would be a great partnership. Thanks to every speaker in this session. It's been a very insightful and exciting content, and we really learned a lot, and we look forward to collaborating with you more in the future. So thanks everyone, and we will see you at the next session after a short break, which will start at in 22 minutes. Is that right? No, in 12 minutes. Okay, 12 minutes. See you then. Thank you.